This is part 3 of Module 1, the Chemistry of Life. In this part, we'll cover acids and bases and also the macromolecules. Acid, bases, buffers, and pH. An acid, by definition, is a proton donor. Let me tell you about a hydrogen ion. Remember, hydrogen has one proton and one electron. If that electron is not present, then it is a hydrogen ion with a positive sign here. It is essentially a proton. So that will be used interchangeably with hydrogen ion, and you'll see hydrogen ion written out, and that is a proton. An acid is a proton donor, a base is a proton acceptor. So this is hydrochloric acid right here, and in water, it's going to dissociate, and it's going to donate that proton or hydrogen ion. This is bicarbonate, and it can collect or accept hydrogen ions to form carbonic acid. pH is a scale from 0 to 14. The lower the number, the more acidic. The higher the number, the more alkaline or basic. What the pH indicates is a concentration of hydrogen ions or protons. The pH is going to be inversely proportional to the concentration of hydrogen ions. That means that when there are a lot of hydrogen ions, concentration high, pH is low. When there are not many hydrogen ions present, the pH is high. So pH of 1 is very acidic, high hydrogen ion concentration. Our bodies maintain a pH of 7.35 to 7.45. There are lots of different what we call buffer systems within our bodies to do that. A buffer system is the combination of a weak acid and a weak base that can either collect or accept protons or hydrogen ions, or give away the hydrogen ions whenever necessary in order to maintain a pH. Remember, the pH is related to the concentration of hydrogen ions. This carbonic acid buffer system is a common buffer system in the body. It's in our bloodstream, it's in our kidneys, it's in our lungs. If the hydrogen ion concentration goes up, well, this equation, you notice these arrows go in both directions, that means it's reversible. This equation goes in this direction, and those hydrogen ions form carbonic acid, which can then convert to carbon dioxide and water. What happens if the hydrogen ion concentration goes down? Well, the opposite happens. This equation goes in this direction, and carbon dioxide and water forms carbonic acid. The carbonic acid then dissociates, and we can then increase the amount of hydrogen ions back to where it should be. This is a buffer. Let's talk about salts. For our purposes in biology, we don't have to worry about a lot of different salts, but a salt is an ionic combination of anion and cations. Remember, anion is negative, cations are positive. You might be able to remember that because the T forms a little plus sign. As far as the anion, there are others, but the main one is going to be the chloride ion. As far as salts, these are the 4 major ones. We have sodium chloride, potassium chloride, magnesium chloride, and calcium chloride. You might ask, well, why are there 2 chlorides with the magnesium and the calcium, but not the sodium and the potassium? Remember, sodium and potassium on the periodic table are in that first column. In the first column, there is 1 electron in the outer shell or valence shell, and that 1 electron will be stolen by the chloride. In the second column, and that's where magnesium and calcium are, there are 2 electrons in the outer shell. So each of those elements or atoms has 2 electrons to give away. It will give 1 to 1 chloride and another to a second chloride, and that's why you have 2 in magnesium chloride and calcium chloride. Let's change gears a little bit, talk about macromolecules. Macro meaning big. So these are large molecules, lipids, carbohydrates, proteins. So let's start with lipids. They're usually nonpolar, meaning they are hydrophobic. They will not dissolve in water. We can talk about triglycerides. I'll show you a picture in a minute. Triglycerides are what we use to store energy. That's the main purpose for triglycerides. Phospholipids. These are what we call amphipathic, and we'll get more into that when we cover that in the textbook. Amphipathic means that there's a polar part and a nonpolar part. So it has part of it dissolves in water and another part that won't dissolve in water. Phospholipids form cell membranes. And then we have steroids, and steroids are produced from cholesterol. Yes, your body needs a certain amount of cholesterol in order to produce these steroids. The steroids make up many of our hormones. I'll show you some pictures of the different chemical structures for a few of these lipids. This is a triglyceride over here on the right-hand side, the triglyceride. How you get a triglyceride is you add a glycerol to 3 fatty acid chains. We'll talk more about fatty acids, but just to give you a general idea, each of these has what we call a tail, lots of carbon and hydrogen. If you remember what I said about carbon bonded to hydrogen, that is nonpolar, and that is why these molecules are nonpolar. They will not dissolve in water. There are not very many polar bonds. They're kind of hidden here on the inside. All right, so this is a triglyceride. This is how we store fats. This is a phospholipid. This is how it's typically depicted. There's a phosphate up here. Phosphate is a phosphorus with some hydrogens attached. This is a one of the what we call polyatomic anions. It has a negative charge here. There's a glycerol, and then there's a couple of fatty acid tails. And this is a way that sometimes these fatty acid tails are depicted as these squiggly lines. At each bend here, there's a carbon. And if you don't see hydrogens attached, there are hydrogens that you have to kind of put in there in your mind. This is just a shorthand way of showing the hydrogen tail. This is a phospholipid. The part up here is hydrophilic, and down here is hydrophobic. So one of the things that these molecules do is the hydrophobic part, which is in the middle here, associate with one another, and then the hydrophilic part down here and up here are on the outside. And these words here, extracellular, this means outside the cell, this is inside the cell. So outside the cell is solution like water. Inside the cell is cytoplasm, also like water. So the polar part is associating with the water, and the nonpolar part is on the inside, and this is depicts what a cell membrane looks like. And it's called a phospholipid bilayer. Carbohydrates. Carbohydrates consist of monomers. A monomer is a molecule that when you put a bunch of them together, you get a polymer. So the monomer for carbohydrates is a monosaccharide. Glucose is an example, but there are other monosaccharides. Some of the polysaccharides that you'll see, starch. If you eat a bunch of macaroni and cheese, bread, that contains starch, which is really just polymers of glucose. So a bunch of glucose molecules stuck together. Glycogen is also a polymer. It is a polysaccharide. A bunch of glucose molecules stuck together. Glycogen is how our body stores glucose. We store it as glycogen in our liver and skeletal muscle. Cellulose is also a bunch of glucose stuck together in a little bit different way. We cannot digest cellulose. It's what we call fiber. Carbohydrates are hydrophilic because they contain mostly polar bonds. And if you look at the molecule of glucose here, you see carbon with oxygen, oxygen with hydrogen, oxygen with hydrogen, oxygen with hydrogen. And as I told you before, those are going to be polar covalent bonds, electrons being unequally shared. A polar molecule like this is going to form hydrogen bonds with water when you put it in water, and that allows it to dissolve. Nucleic acids. The monomer for a nucleic acid is called a nucleotide. I'll show you a picture in a minute. You're probably familiar with DNA and RNA, deoxyribonucleic acid and ribonucleic acid. DNA is double-stranded. These are the polymers. Adenosine triphosphate, or ATP, is also a nucleotide, and it's very important because it's what our cells use for energy, and we'll talk about that in the section devoted to energy. This is DNA, deoxyribonucleic acid, and again, the monomer for this is a nucleotide. I'm going to circle one nucleotide here, and a nucleotide is going to consist of a nitrogenous base, and that's this part of it here, and this particular one's called guanine, a 5-carbon sugar, which is right here, and then a phosphate. So that makes up the nucleotide. That's the monomer, and it gets repeated, although the nitrogenous base may not always be guanine. And here you see one that is thymine and one adenine, and here's one that's cytosine. But these get repeated, and DNA is a double helix, and it forms something like a ladder, and the rungs of the ladder are one nitrogenous base hydrogen bonded to another nitrogenous base. This is adenosine triphosphate, or ATP. Again, there's a nitrogenous base, a sugar, and then there are 3 phosphate groups, hence the triphosphate. And the thing to remember here is that this is a high energy molecule. This is what our cells need in order to perform all sorts of cellular functions. The bond here between the last 2 phosphate groups is a very high energy bond. Key concept in energy is when you make a bond, it takes energy. When you break a bond, it puts out energy. So if you were to break that bond between the last 2 phosphate groups, you get a release of a lot of energy that can then perform cellular function. So we've talked about lipids, we've talked about nucleic acids, we talked about carbohydrates. The last one is proteins. The monomer for a protein is an amino acid. If you put a bunch of amino acids together, you get the polymer, which is the protein if it's greater than 200 amino acids. If it's less than that, we call it a polypeptide. Poly meaning many, P-O-L-Y. Now there are 20 different amino acids. You can string those together in all sorts of different combinations to form the proteins. Proteins have a specific structure, and that structure is important to its function. If the structure is changed, the protein may not function. That brings us to talk about the concept of denaturation. Denaturation changes structure of proteins. An increase in heat or any change in pH. So if you have a really high fever, that can damage or denature proteins, and proteins can be enzymes, which we'll talk about in the next section. Proteins can be part of our cell structure that keeps the cell intact. Proteins have a lot of different functions. But a high temperature is going to start denaturing proteins. Any change in pH, so if you add hydrogen ions or take hydrogen ions away, it's going to disrupt some of the bonding within a protein, and it's going to denature it. So here we have an example of a protein. The different little circles here, different colors, represent the amino acids. And again, you put a bunch of amino acids together, you get a protein. The bond between amino acids happens to be called a peptide bond. Here is a random amino acid. Remember I said there were 20 of them. We'll go into the structure of amino acids more during the semester. And as I said, proteins have specific structures. So if we just talk about the string of amino acids, that's the primary structure, primary protein structure. Then those amino acids can fold onto one another and form different bonds between amino acids, and form what we call alpha helix or a beta pleated sheet, and that's the secondary structure. Then there can be more folding, and you would get a tertiary structure. And sometimes we talk about globular proteins. Enzymes are globular proteins. So that's the tertiary structure. If you put more than one protein together, and that happens, hemoglobin, for instance, is 4 proteins put together, we would call that the quaternary structure. Now again, don't forget, denaturation can change the structure and make the protein non-functional. And here's an example of that. We have normal protein here. It's folded on top of one another. It's globular protein. We're going to denature it, and it's going to lose its function because now it has unfolded. There's denatured protein here. And again, any change in pH or an increase in temperature will do that. There are some other things, but for our purposes, just remember those two. And that's it for Module 1. I'll come back with Module 2 in a separate recording.